I think this is everyone for now, so I'm just going to start with the introduction already. So again, for the people that just joined, I hope you're having a great Tuesday and welcome to PaleoPerks again. And today we're going to listen to a talk by Luz Maria Mejia Ramirez from MARUM in Bremen, and who's going to talk about new coccolithophore and diatom-based geochemical temperature and CO2 indicators, improving climate reconstructions and proxy understanding. So first, a bit about the format of today's seminar, especially if you're new here. First, we're going to do the welcome and announcements. That's what I'm doing right now. Then we'll have Luz Maria's talk. This will last around 30 minutes, followed by a moderated Q&A. And for this, you can send your questions to the questions host, who is Mohammed Salman today. And after that, Luzma has agreed to stay for tea time. So if you want to have a more informal chat with today's speaker, then feel free to stay afterwards. Then before we start, a bit more housekeeping. So PaleoPerks values the participation of all folks interested in the paleosciences. And please remember to abide by our code of conduct during today's webinar. If you're here in the Zoom room, you should have agreed to the code of conduct. If you haven't, then please take a moment to visit the website and read through that. Also, out of respect to the speaker, please keep yourself muted for the duration of the talk. You should not be able to unmute yourself, but if you do find yourself unmuted, please mute yourself again. And as I said before, you can ask questions by chatting to the questions at PaleoPerks host. Then you can ask them via chat. You can also use the raise hand function during the Q&A if you want to ask a question yourself. And also, if you have any technical issues during the talk, you can send questions to the questions at PaleoPerks host. Then we also have closed captions built into Zoom, and you can use the CC button to either hide them or show them. Then finally, we're always looking for more speakers for PaleoPerks. So if you know someone who could give a great talk and is an early career researcher, or if you want to give a talk yourself, you can nominate someone or yourself in the link. This link will also be dropped in the chat. And if you want to give us weekly feedback, we're always looking for some feedback and demographic information to improve PaleoPerks. So you can also fill that out if you want. Will also be dropped in the chat. So that was enough about PaleoPerks. Now a bit more about our speaker, Luz Maria Mejia Ramirez. As I said, she has done her bachelor's in marine biology at the University of Tadeo Lozano in Colombia. She then went on to do her master's in marine biodiversity and conservation at the University of Oviedo in Spain. Then she kind of made the step towards a bit closer to geosciences with a PhD in biogeosciences at the University of Oviedo, again in Spain. She then went on to do a postdoc at ETH Zurich, and currently she is at the MARUM Center for Marine Environmental Sciences in Germany. So with that, Luzma, I would invite you to take over the screen and start your talk.
Okay. Hi, everyone. I'm Luz Maria Mejia. First of all, I want to thank the steering committee of PaleoPerks to have invited me to give this talk. And today I'm bringing you a set of short stories of like new or different kind of temperature and CO2 proxies based both on coccolithophores and diatoms, and how we have used them to improve climate reconstructions and hopefully also improved our understanding of more like well-established proxies as well as our proxies. So first of all, I really like always to start a little bit with my motivation, what brought me here. So as Nina mentioned, I am from Colombia, which is like the uppermost country in South America. And this country is like the most biodiverse country in the world. Okay, so the first one being Brazil, but Brazil is huge. So the first most biodiverse country in the world in terms of area. And I don't know if you watched the Disney movie Encanto a couple of years ago, but it is, as you can see in that picture, it's like full of colors. Colombia is like everything is growing and thriving, and it's just like showing so many colors. And I had the really nice opportunity to see all these colors doing marine biology and there. but also had the opportunity to see, like, firsthand experience how these colors have been disappearing because of climate change. And this is actually why when I was doing my PhD, I decided to accept being kidnapped by geologists and kind of leave those colors in standby, because I really like, for example, sea turtles, corals, everything in the tropics is just boom, and focus more on better understanding climate change. And so I changed those colors and I ended up working with white powder and going around the world with white powder as a Colombian. Uh, so with coccolithophores and diatoms. And you can imagine, for me, that was kind of a shock, because I ended up working with things that you cannot see with your bare eyes. You need microscope. They are dead and they have no colors. They are white. So, but at the same time, I kind of realized that the impact of doing this could be much larger than just focusing on saving one species or one species in one region. And just targeting climate change as a whole could actually help me save as much of those colors as I could. So this is why I made the change. And this is why we do what we do. And I always like to remind everyone this. Because this is like trying to understand how our world, so how our climate and therefore temperature is going to react to the CO2 that we are putting in the atmosphere, by understanding how it was the reaction of the temperature to the CO2 there was during times in the past that we think were similar to what we are going to face in the future, is the only way that we are going to be able to do mitigation strategies or take actions on time to be able to try to stop, not stop, but slow down the losing of these colors and biodiversity, which is what made me fall in love with life. So, yeah. So for that, we need to be able to improve both our temperature and CO2 reconstructions. And just a little bit about CO2. This is a compilation, not the last one, but here you can see as well how it could be looking in the future. Oh, wait, I changed my screen. What did I change? Okay. Yeah. Are you seeing the screen?
Yes,
Okay.
everything looks
Okay.
normal.
Okay, so here you can see that, that in many times we don't really know how much CO2 there was. There could be 400 or there could be 1,000. So it's very uncertain still. And therefore we really need to target more like how much there was, like the magnitude of what there was, so we can know where we are going to go in the future. Like, are we gonna have beach trees in Antarctica or alligators in the Arctic? Like, what can we expect? So now, wait, this is not working. Okay, did it change now?
Yes, now
Okay.
the bottom plot is gone.
Okay, cool. Okay, so now I'm going to focus on temperature. So I like to divide the temperature proxies that we have into like two different types. So the inorganic ones being delta 18O, magnesium, calcium, that are usually or traditionally applied to foraminifera. The main problem about these inorganic proxies is that we have to rely on assumptions of how seawater chemistry was in the past. And sometimes we don't really know how seawater chemistry was in the past. And then we have the organic proxies, like for example, TEX86, which is based on organic molecules produced by archaea. And the main problem about this proxy is that archaea can either live from the very top of the surface ocean to the very bottom of it. So most of the times we have like an uncertainty of where the signal is coming from. And then we have alkenones, which are produced. So these are based on organic molecules that are produced by coccolithophores. And the mainly described problem about this proxy is that it saturates at temperatures around 28, 29 degrees, meaning that if coccolithophores were living and calcifying and doing photosynthesis at 35 degrees, we are never going to be able to measure more than 28, 29 degrees. For me personally, the main problem about these organic proxies is they are solely based on a correlation to temperature. And we don't really know which is the mechanism that is linking them to temperature, why they are being produced, and we also don't know if there are other mechanisms that could be driving variations in these proxies. So I want to use this opportunity to encourage you to take action in contacting more ecologists, biologists, physiologists, because this is the only way we are gonna really understand our proxies, like what it is that what it means of the data that we are producing, because these bugs are actually not that simple at all. Okay, so we have used clumped isotopes in coccolith calcite to try to circumvent some of the problems of other proxies. So first of all, why do we want to use clumped isotopes? Mainly because this is a proxy that we know how it works. It works by measuring the amount of bonds that are made between the rare isotopes of oxygen and carbon, so 18O and 13C. And it is pretty simple because it's only theory based only on thermodynamics. And it's simply like the colder it is, the more clumping we will have. And the warmer it is, the less clumping we will have. So theoretically, if this was like this, we would have like the God proxy, proxy, because we would have a proxy that would be independent of seawater chemistry and would also be independent for any biological overprints or so-called vital effects. But then, like, we now come to Earth and we realize that actually there are some vital effects. Like, for example, we have seen already vital effects in corals. Regarding coccolithophores, it is kind of new. Not a lot of investigations have been done in coccolithophores, but so far so good. There has been 2 studies doing culture studies for clumped isotopes in coccolithophores. So in this graph, you can see in the x-axis, we have large vital effects in both oxygen and carbon isotopes for 3 different species of coccolithophores, culture coccolithophores, but we don't see big differences or differences that can be actually measured in clumped isotopes for these species. And very similar results were found last year by the culture study by Clark et al. So far so good. It should be good to measure coccolithophores, like mixed species of coccolithophores. Okay, so why? Now from coccoliths. Why from coccoliths? Well, because coccolithophores are organisms that are relying on light because they are photosynthetic and therefore they ensure that our signal is coming from the surface. So while foraminifera can go up, can go down, can go and mate there, go and eat there, so their calcite is actually being produced at different depths of the water column, coccolithophores rely on light and therefore they can only be produced in euphotic oceans. And the second reason why we want to use coccolithophores is related to this question. So please just put your microphone on, and if you feel like answering, answer. Otherwise, I'll just wait a couple of seconds and I'll answer. So what do you think these two have in common? So a coccolithophore and a pregnant Shakira.
You can also use the raise hand function or put your answer in the chat if you want to.
Oh, that's gonna be hard for me because I don't know where to. Oh no, I shared screen.
Oh, sorry, I see that Miriam Shuar, I hope I pronounced it correctly, has an answer.
Yeah.
Okay.
Yeah, they're both growing their stuff inside of them.
Yeah.
Like well, coccoliths, they grow new shields within themselves, and Shakira's growing a baby inside of her stomach.
Exactly. Thank you, Miriam. Yeah, so coccoliths are actually done in a very tight and controlled environment inside the cell. And when they are ready to be done, they just pop, they just go out of the cell, and they cover each shield, let's say, each plate with a cape of polysaccharides. So they are actually being protected by polysaccharides. So when they die and they sink, it is much less possible that they are affected by diagenesis compared to, for example, other type of calcite or aragonite. So they are more resistant to diagenesis. So cold biases done related to overgrowth or recalcification are less likely to happen in coccolith calcite. Okay, so this is the first dataset that I wanted to show you. So we measured clumped isotopes in coccoliths from different core tops all over the world. So you can see here them in the map. And in the figure in red, you see the clumped isotope temperatures. In green, we have the alkanone temperatures with exactly the same in exactly the same samples. And then in yellow, we have the sea surface temperatures during the coccolithophore production season. So first of all, we have a latitudinal gradient, which is cool because no one had done that before. But important to mention is that especially for the tropics, we see that clumped isotope temperatures are lower than sea surface temperatures, and also alkanones tend to overestimate sea surface temperatures. So I was thinking, okay, what if coccoliths are not always living at 0 meters? What if they are living a little bit deeper? So what I did is I went through, like in each of these places, geographic places, I went through the water column and calculated the depth at which they would be living if we believe the clumped isotope temperatures. And this is how it looks like. Especially for the tropics, you see that actually coccolithophores seem to not be living at 0 meters, but still within the euphotic zone, which is marked with light blue shading. And this actually is not a new idea of me or anything. This has been already shown by ecologists, biologists that have been done like ship transects, both in the Atlantic and in the Pacific, where they have seen cocospheres, coccoliths, chlorophyll A concentrations, and all these indicators that are higher at, I don't know, depths of even higher than 200 meters. But the question remained whether these cells were actively growing and calcifying and doing photosynthesis, or they were just being accumulated down there. And clumped isotopes seem to be suggesting that they are actually growing and doing calcification at these depths. And you can imagine that if we find that this is true, it will have very big implications for other proxies, like for example, the interpretation we do of alkanones, because we are currently interpreting alkanones and calibrating them to sea surface temperatures, and which, if coccoliths are living deeper, then this would lead to overestimates in temperatures by the alkanone proxy, for example. Okay, so the second dataset that I wanted to show you is this downcore record from the North Atlantic during the last 16 million years. It's site 982. Something to take into account is that for these downcore records, we always have to take into account that there could be some recrystallization over growth that could introduce a cold bias. So we observed the coccoliths using SEM, and we calculated that at least 92% of them were pristine. We also did a model, and there were like the worst case scenario would be like a cold bias of 2 degrees, like worst case scenario. We also need to target samples that are organic matter free, so it doesn't mess up with the measurements. For that, we do extraction with an ACE, and then we use that extracted organic matter to measure also alkanones in the same samples. And then we also do a step of oxidation. So here I'm gonna show you a little video. Maybe I should have taken the sound off. Yeah, here. And you can see this is not any of the samples that I'm going to show you, but you can see that when we have organic matter still in the sample, it is like pretty explosive, which is not the case for this sample. So if you have samples that are white, it's really cool because it's not going to bubble a lot, or you don't expect that it's going to bubble a lot, but it could be kind of explosive. And yeah, and then we use the extract to measure alkanones, and then we use all the different calibrations that are out there, baseline, core top, all the different culture calibrations to calculate SSTs from alkanones and compare them to clumped. And the last thing, and very important thing, is to be sure that our sample are actually containing coccoliths, because if we work with bulk, then we will have fragments of forams or forams itself or a biogenic calcite, like so many things that are actually not coccoliths. So for that, we use centrifugation, settling, and semi-automated microfiltering to get samples that are containing mostly coccoliths. And in this case, there are like at least 90% pure coccoliths. So these are some examples of the separations here. And yeah, so these are the data. In the upper panel, like in warm colors, you can see the alkanone temperature reconstructions using all different calibrations, so core top, base plane, and all the culture calibrations that there are, which, by the way, can lead up to 8 degrees differences, like the different culture calibrations that are for alkanones for our data can lead up to 8 degrees differences. And then in the down part in blue colors, we have the clumped isotope temperatures. So cool thing, we have similar trends. Not so cool thing, we have like a 9-10 degree differences between the two proxies. So there is a huge difference in the magnitude, although the trends not so much, which is a big thing. So we were thinking, okay, obviously our clumped isotope temperature record is wrong because it's the new one, and the well-established one are the alkanones. So we went to see, okay, what could be wrong with our clump record? So we went to see the SEM, and we modeled how much this maximum 8% of non-pristine calcite could mean in terms of cold bias, and we calculated this maximum 2 degrees. So, okay, no 10 degrees, now 8 degrees, but it's still 8 degrees, you know? Then we have very cool, very pure separation, so it's not that. And then on top of that, we have species that are actually producing alkanones. So we couldn't understand why a 10 degrees, 9 degree difference could come from anywhere in our clump record. So we start to think, okay, what happens with the alkanones? So what I did is I went back to the study of the baseline calibration of alkanones, so the Tierney-Tingley study, and I took only a subdata set, so the subdata set of that geographically match my data set of the clumped isotopes in the core tops, because this is the only these are the temperatures I have for clumped, and instead of calibrating their alkanone data to sea surface temperature, I use the temperature at which clumped isotopes are suggesting coccolithophores are calcifying, so at depth and during a specific season of production. And this is the blue curve here. So when we apply this quick and dirty, I'm not saying this is a cool calibration or robust statistical calibration. Of course, the original data set is much better statistically handled, but this is just a quick and dirty calibration only for the data that we have clumped data from. And when we apply this calibration to our alkanone data here, this is what we get, is the green curve now. So our alkanone data has gone down 8, 9 degrees, two magnitudes that are more similar to clumped isotopes. By the way, something also to mention is that the core top sample here, the clumped isotope match much better the sea surface temperature at the site compared to alkanones that are already overestimating the temperature also in the core top. So yeah, you can imagine that this could have huge implications because there is this conundrum in paleoceanography that models are not able to simulate the warmth in the high latitudes during the Miocene or also during other periods. And we tend to think that models are just missing something there. If they don't do well reproducing our proxies, then it means that they are missing something. So if we believe what the alkanones were saying, so both in the late and the middle Miocene, models tend to do well in the tropics. So you can see here that models are doing well in the tropics, but in the high latitudes, if we believe the alkanones, we will have an ocean that is like a soup. So there is no latitudinal temperature gradient. While if we believe the clumped isotopes, which are here in stars and which match much better with the models, then we would have an ocean that is, yes, warmer than what it is right now, but not as warm. So we would have like modest but not extreme polar amplification. And if we prove these at other high latitude sites, it could mean that perhaps our the way we think the high latitudes are going to react to anthropogenic CO2 may be not as catastrophic as we currently think. So it is like it's a thing that we should look into in more detail. And the last data set that I wanted to show you about temperatures are these low resolution coccolith Cenozoic clumped isotope reconstructions. So we use low latitude size from the Pitt transect in the eastern equatorial Pacific here, and high latitudes from south of New Zealand here in 1170, site 1170. So these are some pictures of the separations, which I am really proud of. So we have very pure separations, and there are some examples of the SEMs that I took. So we don't have a lot of problem of recrystallization, but there is a lot of dissolution, especially in the tropical samples, because they are located much deeper. So this is how the record looks like. In yellow, we have the low latitude sites, and in blue, we have the high latitude sites. We still need to measure the rest of the Cenozoic for the high latitude sites. But the cool thing is that it tends to, to show similar trends to what we would expect from the benthic foraminifera delta 18O. The very, I don't know, interesting or not unexpected thing is that the low latitude sites tend to have like very low temperature. So if you compare the core top here, that is like 22 or 23 degrees, and compared to the first sample we have here that we have at 2 million years, around 2 million years, it's like 8 degrees differences. So it is kind of unexpectedly cold. So we thought, okay, maybe there is early recrystallization, but the SEM doesn't seem to show a lot of recrystallization, although there is a lot of dissolution. Also, when we measure strontium calcium, we get numbers that are typical for coccolithophores. It would be like 100 times lower if we have like a biogenic calcite. And then the species that we have are not so, like the species that could be non-photosynthesizers, like for example, these coasters, are not too many to say that, yes, we have like 10 degrees colder temperatures. So far we cannot explain these cold temperatures. And then we were thinking, okay, maybe it's simply that these temperatures could be true and they are tracking the source of upwelling, or the movement of the upwelling, or also tracking changes in habitat depth. So if you have any idea, just come to me. I will be very happy to hear your ideas. And yeah, so now I'm going to shift to CO2 reconstruction. So this is a compilation, the last compilation that we published in 2023. And you can see that we still have huge uncertainties pretty much all over the Cenozoic of how much, of the magnitude of the CO2 during most of the Cenozoic. So we also have to work on this front to be able to constrain better climate sensitivity. So I'm going to introduce a proxy that has been widely used in alkanones, which is the delta 13C. So people usually measure them in these organic molecules that are produced by coccolithophore, so in alkanones, and then they transform this delta 13C to CO2, and I'm going to show you how. But I didn't want to do what everybody does, because that's the way I am, and I went to measure this delta 13C in the organic matter that is trapped inside the frustal, so the silica of the atoms. But for you to understand how this works, I'm going to have to explain you the phytoplankton diffusive theory, which is what is the proxy based on. So diatoms, coccolithophores, and all phytoplankton have a preference for C12. So they really like to take C12 to do their photosynthesis, right? So you can imagine in the past when there was so much CO2 concentrations, by diffusion, they would get very easily a lot of carbon, which means I'm going to introduce now a term here, which is called fractionation. And I just want you to understand it in a very simple way. If the diatom and the coccolithophore can choose to eat what they want, then the fractionation will go up. If they cannot choose, the fractionation will go down. But since we have a lot of CO2 available through diffusion, then they can choose as much as they can, so fractionation will go up. They have this ability to choose, right? Okay, so now I'm gonna introduce our special character. She's called Roger, and this bunny is going to explain to you how this works. So Roger has two types of food. Cookies for bunnies, which is 12C, which is what she likes to eat. And then this dry grass, which is 13C, which is what she doesn't like to eat so much, right? So we do an experiment and we give her a little bit, or kind of a ton of both. And what do you think is going to happen when we give her easily this food? Well, of course, she's going to eat the cookie. So she's going to eat the 12C, which means that she had a lot to choose and therefore she chose. But what happens under very low CO2 concentrations? There is not so much CO2 coming into Roger by diffusion. So she's not just going to sit there and starve, right? She's going to start to eat the dry grass, because she's just going to eat something. She's not going to die. So she starts to incorporate also the 13C, which means that she doesn't have the ability anymore to choose. So fractionation goes down. Also important to consider in this diffusive theory is that size is very important. If we have big cells or like big rabbit, you can imagine we have a lot of volume to feed. So there's going to be a lot of demand. Not so much surface area for CO2 to go by diffusion, and therefore they are going to be able to choose less. While small rabbits or small cells, they have less volume, so less to feed and more surface area that can go through diffusion the CO2. So at exactly the same CO2 levels, we will have less ability to choose when the cells are bigger and more ability to choose when the cells are smaller. So for diatoms, the way we circumvent any problems regarding size is that we separated diatoms of the same size so that we have a constant surface area to volume ratio, and therefore we don't have to make any corrections for size because we already have the same size. For alkanols, this needs to be done because it's organic matter and you cannot separate them for sizes. So yeah, then from this epsilon p, which is the fractionation, we calculate the CO2 concentrations. So this is how the record looks like. I know it's complicated, but I will guide you through. So here in the blue shaded area, like here in the down part, we see alkanon records, like the old alkanon records, where there was no size correction. And actually there were some papers that were published that were saying there was an decoupling from temperature and CO2. So while temperature was going down during the Miocene, CO2 was not changing. And actually, when you take these records and you do the size correction, which is the pink here, this is for alkanols, then you do see that CO2 is actually going down. And therefore, there was no decoupling between temperature and CO2. So here in the empty circles, so here these ones are my diatom records, which don't require any correction for size because we already have the same size, and you can see that the magnitude and trend match pretty well the alkanone records with corrected size. But there is something additional, which are called carbon concentration mechanisms. So this is another complication. So you can imagine we have Roger there and we have tons of dry grass just on her side, but we have a bag with the cookies that is on top of a table and the bag is open. What do you think the rabbit is going to do? I bet the rabbit is going to jump to the table and put her head inside the bag and eat a lot of cookies, even though it costs her some energy to go and jump on top of the table. So this is actually what phytoplankton have developed through time. So they have developed these carbon concentration mechanisms in which they have to put energy. So CO2, so carbon is not coming through diffusion anymore, or not so much because there is so little, but they are using active transport of bicarbonate inside the organism, which means that now they have a lot of carbon to choose, and then fractionation goes up, which is the reverse as you would expect from CO2 only, like from the CO2 diffusive theory only. So when we take into account these carbon concentration mechanisms, which are the squares here, you can see that there is a huge variation in the magnitude of what CO2 could be. So it could be from 400 up to 1000. So these carbon concentration mechanisms need to be constrained so that we are sure that what we are producing is the magnitude that needs to be used by the modelers to do their models, right? Yeah, so this is my conclusion slide. I just want to remind you that we have to work together to try to understand our proxies better, so that we are sure that the data we are producing, we understand, and where it's coming from, so that we don't end up like these poor dinosaurs that were left in the holy ship. Yeah, thank you very much, and I'm happy to take questions. I hope it wasn't too long.
Thank you, Luzma. You were perfect on time. That was really a super interesting talk. Yeah, again, reminder to the audience, if you have any questions, you can send them to the questions@paleoperks host, and then we will handle the rest. Let's see if we already have some questions. We do. Okay, I will post it in the chat. So the first question says, great talk. Do you think our analytical techniques will ever improve to the point that we can do single coccolith clumped isotope measurements?
Oh, I don't think so. Well, we need well, that's one of the main problems, let's say, like one of the main limitations for clumped isotopes, because we tend to need a lot of material so that we can measure a lot, because they are in so little concentrations that we need to do many measurements so that we have decent error bars, which are still pretty big compared to other proxies. And we need, I don't know how millions or billions of coccoliths right now, even though it has improved a lot. Because before, maybe we needed like, I'm not sure, maybe I'm just going to say like a random number, but maybe we needed like 3 milligrams per measurement, and now we need like 100 micrograms. It's still billions of coccoliths there, so I am not sure if, I don't know, with nanosims or something that would be possible. I mean, with nanosims, it's possible to measure other things, like for example, strontium calcium, or we have measured also boron in diatoms. Has also been measured boron, I think, in coccolithophores, but there are like, those are bigger concentrations, so I'm not sure we can measure clumped isotopes for one single coccolith. But I would say it's not that important, because it's not like, for example, for foraminifera, we know that like one chamber is made when they just were born, and then one chamber is made afterwards, and one chamber is made afterwards. Coccolithophores are like, first, less long-lived, also like they are like single crystals, so they tend to be more homogeneous. So I am not sure if that would improve a lot. I think we will have to, focus more like on getting our errors better, because I guess that's more like a limitation.
Okay, thank you. That makes sense. And that's also a good point, that we maybe don't need these super detailed measurements. There is another question in the chat from Arpita, I hope I pronounced it correctly, that states, can we take other associated or related physical parameters like a paired analysis to check if the variation of temperature uncertainty in this case makes sense or not? And she also says, thank you for the great talk.
I'm trying to understand the question. Another physical parameter. No, I don't understand the question. In which data set?
Does the question answer, Arpita? Do you maybe want to ask the question personally? Otherwise, you can type a clarification in the chat as well. But otherwise, we'll move on to the next question for now. I'm sorry.
Maybe you mean like nutrients or... Okay, wait.
Okay. So for example, in this case, any other physical parameter like salinity or something else to check along with the temperature.
Okay, so I'm not sure to which data set she refers to, but okay, so I'm gonna assume it's for like to try to understand where the signal is coming from. So I think ideally it would be really nice to have like a water depth. We have been discussing to do this, and we're gonna try to do it, like water depth measurement, where you know what the temperature is, you know what the nutrients are, you know where the chlorophyll maximum is, and then you can measure clumped isotopes. I guess the most important, and in different areas, obviously, because in some places you're going to find coccoliths that are living up, in some places you're going, or we have the theory that we're going to have coccoliths living deeper. So in different areas to have like water samples and filter a lot of them. But that has like complications because obviously then we have to filter a lot of them because we need a lot of them to measure and a lot of them to do like many replicates of the measurements. But ideally, that would give us the information, like the proof, if this is what we think it is. For the past, I am not sure. I am not sure if another parameter could help us. Like, I don't know if to determine the real, like which magnitude is closer to the reality. I am not really sure.
Thank you. I think that was a very good answer. There is another question in the chat by Miriam Chua, who says, amazing talk. How do you select which samples to use for clumped isotopes? Is there any pre-analysis needed? And can you only use samples with near perfect preservation? Thank you again for a really interesting talk.
Ideally, yes, you would use samples that are well preserved so that you don't have to use so much models of, okay, if the temperature down in the bottom was this, then how much of what percentage of pre-crystallized coccoliths we would need to make our signal go to this temperature, like less modeling and more like trusting your data. But I'm guessing it's possible also to correct for that if we have, like, for example, temperatures from benthic forams, or there, like, now there is a lot being done like clumped in benthic forams. So there is, I guess there is a way to correct for it if we had like a bigger magnitude of potential recrystallization in coccoliths, like less preservation. And the way I screen for which samples I want to analyze, especially now that I cannot analyze as much as I would like to, is so you pre-filter at 11, because then if you see the bulk, then you don't see anything. And then with the smaller than 11, you can already see if you think there is a size fraction that will allow you to get rid of most of the forams and most of the fragments that you think are not coccoliths. Because sometimes you go through all these, I don't know, 1 month process or whatever, and then you get a sample that you just cannot get rid of some fragments from, and then it's like, okay, I am not gonna measure it because these measurements are very expensive. And so like doing a pre-visual analysis of which samples make more sense to measure and which not. This is what I usually do.
Thank you so much for the very comprehensive answer. We have another question from Yanming Huan, who asks, thanks for the great talk. Have you perhaps validated the new CO2 reconstruction method for the glacial interglacial periods as recorded by the ice cores?
Well, as far as I know, no, I'm pretty sure. I think the only publication that has ever been done with diatoms is mine. People, I don't know, because it's more like like the standard method is to measure this or to apply this phytoplankton proxy to alkenones. So for diatoms, we have never aimed to do this. For alkenones, I think it has been done. And I think, at least for glacial interglacial, it seems to work. But don't 100% trust me on this. I am not really sure. The thing is that, so the hypothesis is that if you go, the further in the past you go and the more CO2 you have, there should be less influence of other things that are not CO2, like, for example, these carbon concentration mechanisms. So it should be mostly it should be mostly based on how much CO2 is going through diffusion or not, because they were not being forced, let's say, to evolve these new ways of eating. But then the colder we get, I think the more we have to take into account these new or like this new evolutionary ways of taking up CO2. But I'm not really sure if I am not really sure if I think it has been done, but I'm not really sure if it fits or it doesn't in the interglacial glacial scale.
Thank you. Great answer. Yan Mingwan also says thank you. We have a few more questions. So the next one, let me paste it. It says, no, I don't want to do that. Such a great talk. Thank you. My question is about your new size-limited diatom records. Is the implication here that there may have been a systematic shift in the diatom size through the Miocene that is confounding other records? If so, could you talk a bit about that?
So I am not sure if diatoms have changed through time in their size. I am expecting they have done it, as most of the organisms have done it. But this is not really relevant because we are ensuring that what we are measuring is the same size, regardless of, how can I explain this? Regardless of if they have changed their size through time, we are targeting the same size. I don't know if this is clear. So maybe there were bigger diatoms in the past, as it is the case for coccolithophores, for sure. But we are not targeting them anyways. So we are targeting always the same size. So we don't need to make these corrections. On the other hand, if you do this as most of the people are doing it, which is with alkanones, and you just extract the organics from the sample, then you just have a mixture of, you have no idea what, because you have a mixture of coccolithophores that could be big in the past and smaller and smaller and smaller and smaller, and you cannot make this physical separation, and therefore you have to make the corrections in the data. So yes, it has been found that this that when you don't apply the correction for these alkanone records, these phytoplankton alkanone records, then you do have CO2 reconstructions that are mostly like less varying, like more stable. While when you do the correction in size, then you start to see that it changes. So we don't have to do this with diatoms because we are working on the how do you say, on the physical, on the fossil, so we can just target and see what we are actually measuring. With the organics, you cannot see where they are coming from. I don't know if
Thank you
it
for the
makes sense.
answer. To me, it makes sense, but if the
Okay.
person who asked it has any further questions, please let us know. Then we have a question from Boris Karatsolis, who asks, could there be a non-calcifying bias, which we know are a big percentage of the modern assemblage, and could that explain the offset? Non-calcifying forams thrive in the summer in some cases and could give the warm temperature signal, whereas calcified ones represent winter and therefore colder.
I don't know. So there are many things that could be happening here. But I don't know if this is something I don't know, and I don't know if it's known. I don't know if non-calcifying forms are producing alkanones, because if they were producing alkanones as well, then of course then we would have like a summer bias for the alkanones, and perhaps more like a winter bias for the clumped. Especially if the calibrations are just assuming that they are produced like, because usually they assume it's like a mean annual sea surface temperature, or for the high latitudes it's even worse because it's like a summer sea surface temperature, August to October. But I guess these are things that we don't know. We're just assuming that it is the same species that are producing alkanones, and in this case we have the same species for the downcore record. So because it's like the reticulofenestras and Gephirocapsas that we have by chance, by good luck, we also have this species. So we have the same species. But then, okay, I was thinking, what happens if alkanones are only being produced when there is a lot of light? Then it does make sense to calibrate them to sea surface temperature, because even if these species are living deeper, maybe they are not producing alkanones, but we don't know anything about alkanones. This is the thing. So just by assuming that these species are producing alkenones at whatever depth, because they are these species that produce alkenones and not other species, we should assume that the temperatures should be produced at the same time as these same species are being produced in calcite. So as long as we don't have so much more information about alkenones, what they are being formed for, why, like what is the pathway, what they are being used for, I guess it's hard to tell if there is a summer bias or a cold bias, except for that whatever we have a bloom or not have a bloom.
Thank you. Again, it makes sense. Yeah, Boris also says, thanks, this is very cool research, and there are many things still to discover, which I fully agree with. So far, we have one more question. Again, reminder to the audience, please send your questions to the questions host if you have any. This anonymous question asks, have you also worked with Mesozoic data, or are you planning to do so? And a somewhat related question, could you extract the coccoliths from hard carbonaceous rocks somehow?
You're tricking me here. It took me ages to try to understand there was something before 100 years ago. Yeah, I guess you can extract coccoliths from hard rocks. I am guessing they're likely gonna be more subject to diagenesis though. And I have a coworker who is focusing on Cretaceous coccoliths, or he will focus on Cretaceous coccoliths. But so far we have only done Cenozoic samples or measured Cenozoic samples.
That makes sense, but again, that's maybe something for the future.
Yep.
Would be very cool. Okay, we have one more question that came in, also anonymous, that asks, amazing talk. Is there any effect of dissolution on coccoliths' temperature reconstruction as they live mostly near the surface, so they can be more prone to dissolution at deeper depths?
I don't think they can be more prone to dissolution at deeper depths compared to other types of calcite, but yeah, this is something we don't know, and we don't know even for other types of more studied organisms like forams. Dissolution, in theory, especially for a type of crystal like are the coccoliths, which are like one sole crystal, we expect not to be differences if, I don't know, if I don't have anything round here. Okay, if this is a coccolith and it starts to dissolve in the edges, so we don't expect that the fact that we are taking out pieces of coccolith in the edges is going to change the clumped isotope concentration, because it's like a crystal that is being formed inside the cell, and it's homogeneous and it just pops out. So we have, although we cannot measure a single coccolith for clumped isotopes, there has been measurements like, for example, strontium calcium, and there is no difference between one part or the other of the coccolith. So yes, this is something we need to test, I guess, like doing some experiments of dissolution. It hasn't been done, but I would also not expect to have a big impact, or I don't see how it could have an impact.
Thank you. Yeah, that makes sense. With that, we are out of questions for now, and we are also very close to the top of the hour. So, Luzma, I want to thank you again for a really great talk, and also really thank you for the audience for the great questions. Before we move into tea time, and some of you might leave, I have a few announcements to share. Yeah, so I was saying then thanks everyone for joining us. Thank Luzma for the great talk. I hope to see you again next week, August 12th, same place, same time, for a talk by Franziska Blattmann of the University of Aarhus in Denmark, who will be talking about early Triassic flames and feedbacks, exploring wildfire climate and ecosystem shifts. So we go a bit further back in time. And again, we would really appreciate it if you can fill out the weekly feedback form or nominate the speaker. We will again drop the links for that in the chat. And with that, the people that want to stay for tea time are free to stay. So tea time is again kind of an informal chat where everyone can turn their cameras on and speak freely with a speaker. It will also not be recorded. So as icebreaker question, we have a question: do you have a favorite proxy? And if yes, what is it? So before we do that, we'll have a little break of around 2 minutes, so you can turn off your screen, your camera, and then I'll see you in 2 minutes. For those of you that are not staying, I hope you have a great rest of your week. And for those of you that are staying, I'll see you in 2 minutes.